All right, next up is the cytoskeleton. The cytoskeleton is going to be a network that provides shape and support for the cell. It's going to help maintain the shape of the cell. It's also going to be responsible for allowing that cell to move. It's going to help anchor organelles in place, keep that nucleus from sliding all over the place, going to help kind of keep organelles where they need to be. It's also going to allow the cell and organelles to move around. It's going to form tracks for organelles to move along. You can kind of think of these as highways for transporting from one side of the cell to another. They're also going to help move chromosomes during cellular division. The cytoskeleton is going to be composed of thick microtubules, intermediate filaments, and thin filaments. Thin microfilaments. And there are going to be two main parts that we would like to talk about. The centriole. The centriole is going to be the microtubule organizing center. It's located near the nucleus. What we mean by this is the microtubule organizing center. Microtubules are going to play a very important role in cellular division. I don't know if you've heard about or studied mitosis before, but the cytoskeleton comes into play a very important role during cellular division. And all of that is organized and orchestrated by the centriole. Microtubules and microfilaments disassemble and reassemble. Intermediate filaments tend to be more permanent. So intermediate filaments are going to give sort of permanent structure and shape to the cytoskeleton. Microtubules, microfilaments, those are going to come around whenever we need them. So it's kind of like picture yourself sitting there in your chair as just a great big round circular blob. And what if you saw sitting next to you on the table was a sandwich and you're like, ooh, I want to eat that sandwich. So you know what your body is going to do if you're a cell? You're going to suddenly magically grow an arm. You're going to grow an arm, you're going to extend out, you're going to build your skeleton so that way you can reach out, grab that sandwich, put it in your mouth, and once you don't need that arm anymore, you're going to disassemble your arm. Your arm is just going to kind of dissolve away and merge back into the giant round circular blob that you are. So there you go. This is something that is very different about the cytoskeleton as opposed to the human skeleton. The cytoskeleton is very dynamic. It builds up, it assembles, it disassembles, it does whatever it needs to, or whatever is required in that moment. So it changes its structure to match its particular function in that moment. So here's just an example of some of these. Here's our centriole. It's going to be made up of 9 sets of triplet microtubules. So here's the microtubule, microtubule, microtubule. We have 9 total sets of these triplets of this organizing center known as the centriole. And we're going to spend more time on the centrioles a little bit later on once we start discussing cellular replication. To branch out just a little bit, or to talk just a little bit more about those microtubules, they're going to be made up of the protein known as tubulin. Yes, as we said before, the scientists are an incredibly creative bunch when it comes to naming things. The microtubules are made of the protein tubulin. They are ultimately going to be responsible for the structure and the movement of cilia and flagella. Cilia and flagella are going to be our sort of motor structures. They are going to be responsible for highly motile cells and how their ability to move around. Cilia are going to be numerous short extensions in a cell that move back and forth, back and forth. Sometimes they're going to be used for the movement of the cell itself. Sometimes they're going to be used for the movement of other things. You can kind of think of them as like oars on a ship or on a rowing team. There's lots of short little oars. They're going to work together, moving back and forth, up and down to either propel that cell through whatever solution it is in, or they can actually be used to propel solution. What would happen, do you think, if you were to sit on a dock and take those oars and start paddling water? Or if you were to stick an oar into a pool and start paddling the water in one direction? You're not going to make the dock go round. You're not going to make the edge of the pool suddenly move. You are permanently grounded. That dock is grounded. But what's going to happen is the water is going to start to spin, or the water is going to start to move. I don't know, when I was little, this used to be our favorite thing that we would do in pools. We would make a whirlpool where we would start pushing and paddling the water in a circle, and the water would move. It would circulate like a whirlpool. And so that's what's going to happen here. If we have cells that are grounded, they are attached to a basal membrane, cells like the cells lining your respiratory tract, they are going to be grounded. However, they'll have cilia on the surface that are going to beat back and forth, back and forth, back and forth. And this is going to cause liquid to wash over or to move past those cells. In the case of your respiratory system, we have cilia that line the cells in your lungs. Your lung epithelial cells have these cilia that beat back and forth. They're going to wash that mucus over your cells. And this is very important. This is how we get oxygen in, carbon dioxide out. This is how we filter dust particles out. So this mucus that's found, that's lining your respiratory tract, is very, very important. It's how we get oxygen from the outside world into our cells, is by washing that oxygen, or taking that oxygen-rich mucus and washing it across your cells. So the cilia that are found in your lung epithelial cells are incredibly important. We're going to come back to this over and over again. In the case of cystic fibrosis, we end up with, instead of a nice thin mucosal layer, we end up with a thick sticky mucus that prevents the cilia from beating properly. It prevents the cilia from moving that mucus back and forth, and it makes it very difficult to breathe. So there you go. Cilia, very important. Flagella are going to be longer than cilia, and they're going to move in an undulating manner, and there's usually going to be only one flagella per cell. This is going to function more like a motor. So it's going to move and propel from the back of the cell, pushing it forward. Great example of these in humans. They are only going to be found on sperm cells. There are many, many cells out there that have flagella, but in a human being, our only cell that actually has a flagella is going to be found on sperm cells. So here's an example of what that looks like. Here's a lung epithelial cell. Here are the cilia that are moving back and forth to beat and move that liquid along. Here we have sperm cells. Sperm cells have these long flagella extended out the back that are going to propel them and allow them to move. Here we have a cross-section of cilia. So you can see the microtubules that are found inside of those cilia and that are allowing them to beat and to move. All right, and that is it for our discussion of the cytoskeleton. Next up, we're going to be talking about cellular respiration and fermentation in the generation of ATP.